Welcome students, we are here going to start the new lesson, Microbes in Human Welfare. My previous class on this topic, which took place yesterday, has described a lot on this topic, but this video is the first video relating to this lesson. Microscopic plants and animals are the major component of biological system on this earth. According to your previous class information and knowledge, the diversity of living organism that you have read in class 11, you must be remembering the kingdom to which these organisms belong. It is the Monera and Protista, the two prime kingdoms to which these microorganisms belong. You must be remembering yeast. Yeast is a unicellular fungi, and that is considered or placed under the fungi, but it is a microorganism. So fungi is one another major group to which these microorganisms belong. As far as the various habitats where these microorganisms can exist, it may be the soil, water, air, or inside our body, the body of animals and plants. And the habitat where these organisms can be found may be the thermal vent, where the temperature may be as high as 100 degrees Celsius, deep inside the soil under the layer of snow several meters thick, in the highly acidic environment, etc. Microbes of various types are the protozoa, bacteria, fungi, microscopic plants, viruses, viroids, and prions. These are the various types of these microorganisms. Microbes like bacteria and many fungi can be grown in nutritive media to form colonies, and that can be seen with the naked eye, or sometimes we need to observe it microscopically. In chapter 8, we have read about microbes that cause a large number of diseases in humans. They also cause diseases in animals and plants, but this should not make you think that all microbes are harmful. Several microbes are useful to man in diverse ways. Some of the most important contributions of microbes in human welfare are discussed in this chapter. Uh, you have read regarding the beneficial aspects of these microorganisms in your previous junior classes also. Here we shall discuss lactic acid bacteria with a prime focus. Lactic acid bacteria, as the name is indicating, it produces lactic acid during its growth. Lactic acid bacteria produce acid that coagulate, partially digest the milk protein. A small amount of curd added to the fresh milk as inoculum or starter contain millions of lactic acid bacteria, which at suitable temperature multiply. Not only temperature, the microbial growth requires a suitable temperature. Apart from this, it requires the nutritive media also in order to get energy. At the same time, it requires an ideal pH also for its healthy growth, and water is one essential component that requires microbial growth. So temperature, pH, nutritive material, and these are the conditions which require the microbial growth. So suitable temperature, these lactic acid bacteria multiply and convert milk into curd, which also improves its nutritional quality by increasing vitamin B12. In our stomach too, the lactic acid bacteria plays a very beneficial role in checking the disease causing organism. The dough which is used for making food such as dosa, idli is also fermented by bacteria. The puffed up appearance of dough is due to the production of CO2. This CO2 is produced during the course of its metabolic activity. It is, in fact, the metabolic waste that is released during the course, and this release of CO2 is virtually the sole fact that we require or it is useful in the preparation of various confectionaries. Similarly, the dough which is used for making bread is fermented using baker's yeast, that is Saccharomyces cerevisiae. A number of traditional drink and foods are also made by fermentation by the microbes. Toddy. In our vernacular language, this toddy is named as tadi. This toddy is a traditional drink of some part of the southern India and made by fermenting the sap of palm tree. Microbes are also used to ferment fish, soybean, and bamboo shoots to make food. Cheese is one of the oldest food item in which microbes were used. Different varieties of cheese are known by their characteristic texture, flavor, taste, and specificity coming from microbes used. Swiss cheese are due to the production of large amount of CO2 by the bacterium named Propionibacterium shermani. The Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. Microbes in industrial products. Fermentation is the process by which these microbes convert the sugar into ethyl alcohol. This is called alcoholic fermentation, and beverage industry that produces alcoholic drink is one of the major sectors as far as the use of microorganisms in the beverage industry is concerned. Production of wine, beer, whiskey, brandy, rum, these are the examples where the production of such drinks is possible by the contribution of these microorganisms. This Saccharomyces cerevisiae is used for bread making and commonly called brewer's yeast. It used for fermenting malted cereals and fruit juice to produce ethanol. Depending upon the type of raw material used for fermentation and type of processing, without distillation, different types of alcoholic drinks are obtained. Wine and beer are produced without distillation, whereas whiskey and brandy and rum are produced by distillation of the fermented broth. Here is a question: what is the reason that the alcoholic drink like wine and beer is produced without distillation, whereas whiskey, brandy, and rum it is produced by the distillation of fermented broth? The fact is that while the production of whiskey, brandy, and rum, it requires to have a greater percentage of alcohol, and this greater percentage of alcohol can be possible only with the help of distillation, whereas in case of wine and beer there is a lower concentration of alcohol in it, and hence it doesn't require any distillation. The next important product that we are obtaining, or it is I think the major breakthrough in the field of the scientific exploration regarding microbes. Antibiotics are the substances which was very first time, credit goes to Alexander Fleming, who for the first time invented penicillin from a fungi that was named as Penicillium notatum in the year 1929. And thereafter, lot of antibiotics that brought about a revolution in the field of medical science for the treatment of various diseases. These antibiotics played a crucial role. Alexander Fleming, while working on Staphylococci bacteria, once observed a mold growing in one of his unwashed culture plates, around which Staphylococci could not grow. He found that it was due to the chemical produced by the mold, and he named it penicillin after the mold Penicillium notatum. However, its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain, and Florey were awarded Nobel Prize in 1945 for his discovery. Various diseases that antibiotics could treat in the long run were plague, whooping cough, diphtheria, leprosy, etc., which killed the microbes or stopped their growth and saved the life of millions of people. That's all in this video. We shall discuss the remaining part in the next video. Okay, thank you.